PCR, again, many of you are familiar with PCR, but my focus will be mainly on the principles of how do you amplify a gene, what kind of considerations do you do for a gene. As much as possible, we'll do that, and maybe we'll take a break. Okay, what do we mean by gene amplification? Again, very quickly, we learn it from each other. You extract nucleic acid material. In many cases, by the way, there are some genotyping methods you don't start by extraction. And I'm sure Riley will cover this afternoon. If you do, for example, pulsatile gene electrophoresis, the first step is not extraction. You can start from the bacterial cell many steps. But in a classical, typical molecular methods and genotyping methods, the first step is extraction. You extracted already. We discussed in the last 5 minutes. Your goal is to amplify a specific gene segment in that genome. Okay? You might be looking at, let's say, Shiga toxin gene of E. coli 0157, or MEK-A gene of methicillin-resistant Staph aureus. But you have to have some prior knowledge of the gene. Your goal is to amplify that gene using, typically, it's called a thermocycler. Polymerase chain reaction is the method. And once you amplify that gene, you can detect it visually using a gel at an expected size, right? Many of you are familiar with this. And then you can do further analysis. You can use it for sequencing, for cloning, other purposes, or specific genotyping for answer epidemiology question, okay? So we'll look at step by step, but again, this is just the same concept here. What are the steps in PCR? I think most of you are familiar now, other voices. What are the steps? There are 3 essential steps. What is first step? Denaturation, right? So I hope everybody is clear that DNA is double-stranded, right? Double-stranded DNA can be denatured, okay? So in PCR, it is a thermocycling method. So using a 94, 95 degrees centigrade, you can denature DNA, right? Let me ask you a totally different question, just curious on your science background. But if I want to denature DNA, but not for heating, for different purpose, is there any other way to denature DNA other than heating? Chemical, what chemical? Choose one. Yeah, alkaline. So if DNA is treated with alkaline, sodium hydroxide or something, it denatures as well. Just again, that is for different molecular purpose. You need to know that. Like in alkaline lysis I mentioned earlier in plasmid. Yeah, you use alkaline-based method. But anyway, let's get back to PCR. It's a thermocycling method. The first step is denature. Heat-treated denature. The second step is annealing. Annealing what? Annealing a primer. Okay, and we'll look at what primers are, but everybody is clear what are primers? Short oligo sequences that complement to the gene you are targeting. You are looking at specific gene, so you develop an oligo sequence, a short sequence that's called primers that complement with your gene. So anneal is attaching your primers. What you see here in black is your primers annealed to each of the strands. And the third and essential step, and all of these are essential of course, is extension. You extend or polymerize the gene. Okay, it's done at 72 degrees centigrade, and there is specific enzyme, Taq polymerase. Okay, many of you know that, right? Okay, so denature, anneal, extend. What is annealing temperature? And then we'll look at that. What is the temperature for annealing? 50, 60, depends. Okay, we'll address that. It depends, okay? It depends. If you saw a very nice article on Science last week which used, 58, you don't just adopt it for your study. right? It depends on the melting temperature of your primers, and we'll look at that a little bit in detail. Okay, so typically in amplification, again, it's a polymerase, it's a chain reaction. These three steps, denature, anneal, extend. Denature, anneal, extend goes for several cycles, typically 30 cycles or 40 cycles. Okay, then you get a geometrically amplified gene to be able to see it in your gel electrophoresis. Okay, just if you take example of 30 cycles, the gene will be amplified more than 1 billion times, as you can see here. Okay. So many folds geometrically amplified. Okay, and okay, how do you design and how do you know your PCR product size? That's essential thing, again, especially not with a real-time PCR, on the traditional classical PCRs that we all use still for, and in the future we'll use it as well, is your product size or amplicon. The amplicon size is important, right? So what size should I expect if I am amplifying gigatoxin gene from E. coli 0157? The size I expect needs to be defined, right? If I get on the wrong size, that most likely is not the gene I'm looking for. It might be non-specific or it might be some different gene. So you need to have some expected product size. How do you determine that? You look at your primers, your forward primer and reverse primer on your gene. Where do they anneal? And that, you know, besides the flanking region, that's the middle part. Let's say it is 500 base pair, is your expected size. Right? So that's an important thing to know in PCR, so that you are sure that what you are detecting is the genes that you are expecting. Clear? Okay. Okay, guys are really advanced. I think this is clear. Okay, what are the components of PCR? I'm sure you all, essentially, you know, most of you or almost all of you know that. The template, that your nucleic acid material you extracted earlier, it could be DNA, it could also be RNA. A reverse transcriptase PCR, that's, we don't cover it today, but RT-PCR is one. Again, there is a lecture on the website. You need two primers, forward primer for your gene and reverse primer that complements on each of the DNA strands. You need your building blocks, the DNTPs, DATP, DCTP, DGTP, DTTP. Those are the building blocks for one stack polymerizes. You need some buffer to stabilize all your reaction. You know, remember 30, 40 cycles of PCR are happening in this tube. Yeah. And then, very important is the polymerase enzyme, the TAC. It's a thermostable one that we use so that you can run through the 94 and denaturation and all that cycle. These are the important components, and you are familiar. And then there are various things you need to consider when you use enzymes, depending on your purpose, you know, to make it high fidelity, high accuracy, highly efficient PCR. You have to consider, you know, you have different choices of polymerase, TAC polymerase type of enzymes. There is high fidelity versions and different considerations you need to do. Okay. The classical TAC, it's from Thermus aquaticus. When PCR came into this world, I think 1986, Karimoulis, essentially the method was there is a chain reaction. He came up with denaturation, annealing, extension to amplify genes million, billion times, which is a great principle. There was one problem at the time, because the denaturation temperature also denatures proteins, and enzymes, polymerase are also proteins, right? So each time, every cycle, you have to add polymerase enzymes. Second cycle, you open, I don't know how they did it at the time, but it must be very tedious. You add enzyme. Third cycle, you add enzyme. That was a very awkward place, but can you imagine doing that? It would be really boring. But then, since the discovery of this TAC polymerase from Thermus aquaticus, which resists that temperature and stays stable, really it revolutionized the PCR further. So you just add all your components, including your polymerase, you run all 30 cycles, like we do it today. So it's really a big advantage for in the PCR world.